Human Genome Project. What is Human Genome Project? That what is it exactly? What is Human Genome Project? The first question raises here. What is Human Genome Project? What is genome? So that we have to answer all the questions first of all. Then after we need to move with a topic that Human Genome Project is a mega project. It was a mega project because it was completed actually. It was an international effort formally began in October 1990. It has begun October 1990, expected to be completed over in 15 years. Okay, that means 2005 their target, but actually completed in 2003, 2 years before, because of the advancement in the technology, that it was completed before itself. That because advancement in the technology, it was completed before, 2 years before. So began in October 2003 and was completed in 1990, October 1990, and was completed in 13 years project. It became then 15 years project is 13 years project. Who organized this particular Human Genome Project? US Department of Energy. Human Genome Project is organized by US Department of Energy. US Department of Energy and the National Institute of Health. US Department of Energy and the National Institute of Health. These people has begun this particular project. National Institute of Health has started this particular project organized by US Department of Energy and the National Institute of Health. During the early years of HGB, the Welcome Trust UK, every line is important in Neet point of view, every line, because very small area and almost all we are having a particular representation every time. So that's why every line that gives a technical information is very important for us. The Welcome Trust UK, United Kingdom, became a major partner, and additional contributions came from Japan, France, Germany, China. They are also contributed in this particular project, Human Genome Project. The project was almost completed by 2003, that we started in 1990 and completed in 2003. Knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose and treat, and someday hopefully prevent thousands of disorders. The project aim is to make the treatment of the disorder, genetic disorders, whatever hemophilia, color blindness, DMD, incontinentia pigmenti, and chromosomal disorders, Klinefelter, Turner, Downs, Edward, Pride-Chart, Patau syndrome. Likewise, various disorders, how they are coming, why they are coming, and what makes them to come, how to treat, all that answers are there in Human Genome Project. So that it was a beginning. That means in future time when that this knowledge of Human Genome Project is going to open that a new era in biology, that in the treatment of medicinal treatment, genetical treatment, that particular treatment of the chromosomes is going to be expected over in the coming days. Okay, along with the Human Genome Project, a rapid development of new area in biology is also followed over, that is bioinformatics, application of computers, the applications of computers, the data, means whatever the project information, when we are going to store the data, along with the Human Genome Project, bioinformatics also, a rapid development of bioinformatics also observed over. So these are the primary aspects here, that what is the Human Genome Project. We have to see that a project which began in October 1990 and expected to be completed over in 2005, but before 2 years it was completed 2003, and organized by US Department of Energy and the National Institute of Health. Then co-partner Welcome Trust UK, and along with that some other countries have having their own participation. Their contribution is there: Japan, France, Germany, China, and others. We have an Indian participation also. Then, besides providing clues to understanding the human biology, learning about non-human organisms' DNA sequences also very important here, because when we are doing experiments on the Drosophila melanogaster, so knowing the genetic map of Drosophila melanogaster is also very important for us, isn't it? That Drosophila melanogaster means human genome project not only aimed on the human being, so that we that aimed over on other organism, many non-human model organisms. Among them, as we already said, Drosophila is the Cinderella of genetics. From insects, Drosophila, and bacteria, a bacteria from bacteria Escherichia coli, genome is also studied over, and yeast, bacteria, yeast, Drosophila, and from the plants, the rice and Arabidopsis. Rice plant and Arabidopsis thaliana is the plant. Arabidopsis, we see. Let's write Arabidopsis. From the plants, Arabidopsis thaliana, Arabidopsis plant. And one of the good example for linkage, actually, Arabidopsis thaliana, that when in the test cross 7 is to 1 is to 1 is to 7 ratio comes, which is not like Mendel experiments has done on Arabidopsis thaliana itself, linkage experiment. But human genome project not only aimed on the human reading of the decoding of the genome of the human being, also focused on some bacteria, some yeast and insect wise, Drosophila, and a nematode, Caenorhabditis elegans, and a nematode, Caenorhabditis. We can say it's a nematode form nematode elegans, Caenorhabditis elegans, it being a nematode but not a parasite it is. It's a free-living, non-pathogenic nematode. Experiments has done on them also. So, to be remembered, genomes of many non-human model organisms such as bacteria, yeast, Caenorhabditis elegans, a free-living, non-pathogenic nematode, and Drosophila, plants like rice and Arabidopsis thaliana is also being sequenced. In a way, they helped the progress of human genome project also. Okay, so the DNA sequences can lead to an understanding of their natural capabilities that can be applied toward the solving challenges in healthcare, agriculture, energy production, environmental remediation. All these are the benefits we can say here, that application of the knowledge of the genome, human genome project. Okay, we'll discuss time being. Then first of all, let's come with the goals of HGP, the goals of human genome project. Goals of human genome project. So the goals of human genome project include identifying approximately 30,000 genes which are located in human DNA. As a keyword I am writing here, it's better you make as a note making in the form of keywords even. That means keywords are very important here. So by looking towards what is a keyword, by looking towards a word, if you are able to say, we can say that is keyword. So this is keyword. Identify all the approximately 30,000 genes in human DNA. That was a goal actually. That was the goal of human genome project. And next, the second, that determining the sequences of the 3 billion, 30,000 genes, 30,000 genes, and 3 billion nucleotide base sequences. 3 billion nucleotide base pairs that make up human DNA, any human DNA. 3 billion nucleotide, reading the 3 billion, determining the sequences of 3 billion nucleotide base pairs. Okay, next. So these are all going to be math the following. The number of chromosomes on the first chromosome, 2,968 genes on them. Y chromosome is having 231 genes. All the issues are going to be. That's why I'm writing these calculations. Anywhere data is not important, but here the data is going to be very important. 30,000. What is 30,000? That 30,000 genes which can make up the human DNA. Then what is 3 billion? The total number of nucleotide base pairs estimated in the human genome are 3 billion. Okay. And improving the tools for data analysis, that is knowledge of bioinformatics is required for this, and addressing ethical, legal, social issues. We can say simply like this. Addressing ethical, legal, social issues that may arise from the project. These are all said to be the goals of HGP. I'm showing in the form of keywords to you. Okay. So 30,000 genes and 3 billion, 30,000 genes are estimated actually. 30,000 genes and 3 billion nucleotide base pairs, and the data analysis, that may improving the tools for the data analysis, and that addressing that particular ethical, legal, social issues which are raised over during this particular the genome project when it is running. Okay. Then next, what is genome? First of all, let us see what is genome. Then after we'll move forward. What is genome? Genome, we can define genome as haploid set of chromosomes. We can say like this: haploid set of chromosomes. We say that genome, haploid set of chromosomes. A genome represents haploid set, not the total. That means in case of human being, 23 pairs of chromosomes are present. Every pair that is homologous to their the particular another pair. I said this as I named this as maternal and paternal, if you remember. Every pair, one is came from mother and one came from father. We said it as maternal and paternal. So that in our general language, I said here that pair of chapels. We should not compare, but we compared it. Then if imagine if there are 30 people in the classroom, that 30 people means 30 pairs of chapels. We can say 30 pairs of chapels. So for easy understanding, I am using this, that Mendel may excuse us, that what is genome, not 30 pairs or 60 chapels. Among each pair will take one, because your left chapel is homologous to the right chapel, isn't it? So that's why I am taking only one side of the chapel. That we say genome. That means haploid set of chromosomes is called genome. But here, that an interesting thing you can see here. In case of female, 22 pairs are autosomes and one pair is allosome. Male also same. In case of male, 22 pairs of autosomes and one pair of allosome is present in case of male. But there is a genome difference here in that male and female. What is that genome difference? Let us see here that interesting thing. From this 22 pairs, I'll take 22. And from this one, I'll take 1. 21. 22 plus 1. How many will come? 23 chromosomes will come. So, what is genome in female? We say 23. Let us see in male. 22 pairs. From each pair will take 1. That means 22. But you can't take X or Y. You have to take both, isn't it? In case of male, X and Y are completely different. Completely means somehow. That means some very few genes are having both an X and Y. Some are only an X, some are only an Y also there, isn't it? So that's why genome, the meaning in case of male is 24. Male genome 24, female genome 23, because the last pair allosome is same in female. That's why we are taking only 23. But in case of male, that makes some interesting issue, that X and Y are different. That's why 24. So I think you got the clarity actually what is genome. What is genome? What is genome? Haploid set of chromosome. How many in the male? 24. How many in the female? 23. So just you remind that. That's what your general knowledge. And interesting thing even. Next, let's start. Methodologies which are involved in this particular. Methodologies involved in the process of genome project. Methodologies which are involved here. Let's go through them. The methodologies which are involved. The methods involve two major approaches. Here, the methods involve two major approaches. Let me change the color. Here we have two major approaches. which is a frequent question in Neet, always, because there is no much information in our textbook. Let us see one after another. The one approach is focused on identifying all the genes that expressed as RNA, which we say them as expressed sequential tags. See this, very important. Expressed sequential tags. I'm going to explain the first information. I'm trying to explain the focusing on the text information. Then after, I'll give some little more information, the vast information which are involved in this particular, that the various kinds of things involved. I'll show you in the form of chart. Some it is there in text, some it is not there, but we prepared a chart here to give a complete idea on the genome project and what are the results of the genome project. But till the time that let us focus here only. First of all, expressed sequential tags. Okay, expressed sequential tags. What is expressed sequential tag? It was identifying all the genes that expressed as RNA. Let me take an example. We don't have much information in text. That's why it is very difficult to understand. So that's why focus here. There is a nucleus, and this is the genome which is present inside the chromosomes. So first, where they focused actually, we know that particular story, DNA transcription. DNA transcription, which makes the formation of mRNA. mRNA will come out through the nuclear pores, and we learned this story in cell biology. This mRNA will come. This mRNA will come, and excuse me. This mRNA is going to be read by ribosomal RNA, that all the issues we are talking about. So that mRNA itself came out, isn't it? From the DNA transcription, that mRNA is formed, and that particular mRNA itself came out for that protein synthesis process. That what do you say? So these mRNA we are separating. That is one of the particular project that expressed sequential tags. We can say what we defined as, according to the textbook, that identifying all the genes that expressed as RNA. What we do, we'll do the reverse transcription and we'll make the new DNA here. We can, collection of this is so easy here because it came out from the nucleus. Okay, so that is one of the concept here. Then expressed sequential tags. We can say that as expressed sequential tags. Okay. Then just focus here. Just focus. Say that sinking down is going to be a little bit difficult. I am taking my own time. Identifying all the genes that are expressed as RNA are called expressed sequential tags. That is one of the approach. And the second approach is sequence annotation. Sequence annotation. The second approach, we can say the sequence annotation. We can call it as, what is sequence annotation? Let us see what is sequence annotation. Let us see first of all that to know the sequence annotation, let me show you first of all what is the difference between the sequence annotation, to know that, to coding and uncoding, that both things that you have to coding sequences, non-coding sequences, you need to know the information about them. That means whatever we said, 30,000 genes and 3 billion nucleotide base pairs which are present in the human being, all they are not coding sequences. Some non-coding sequences also present. Simply here, I don't much, I'll go here. When you watch a movie in the theater, the length of the movie will be like this, the first box, this movie in theater. But when you watch a blockbuster movie, so-called blockbuster movie in the television, the length of the movie will be like this. Isn't it? Why? Why this much length? So simple. They will give 2 minutes movie and 20 minutes advertisements. This is the advertisement area. Soap konandi, adi konandi, idi konandi అని చెప్తాడు. మళ్ళీ they show you ఒక 5 minutes movie, then ads, then movie, then ad. That means a 2.5 hour movie based on the the people interest, 5 to 6 hours, they'll make it over. Okay, people also will look towards that kind of movies with great patience. After all, that watching that advertisements also. Okay, so our genome is not theater movie. Our genome is that TV display, that TV movie, television movie, a movie which is played in the television. How? How? That means if you take the human genome, a chromosomal content of the human, we are not going to say here that every nucleotide base pair which is present in this particular area, code for something. Okay, some non-coding sequences are also there, enhancers, promoters, regulatory sequences, mini satellite, micro satellites. No need all these names for you, just for I am saying that various other names are also present here. Various other names, other non-coding sequences are also present here. So that we are talking about, that some coding sequences are there. In between, some advertisements are also there, which that are not coding for at all. If you remember in the molecular basis of inheritance, in this chapter, in the beginning, you may learn over exons and introns. One of them, that who are coding for amino acid, who do not code for amino acid. You learned it, right, isn't it? So that's what I am talking about. So at the time in sequence annotation, first of all, we'll read all coding, non-coding sequences, means we read both exons and introns. That exons who do not code for amino acid, introns who code for amino acid. Am I correct? Exons and introns, you learn in molecular biology, molecular basis of inheritance. This is the zoology part. And in the botany part, you learn that coding for amino acid, do not code for amino acid. So in that, not only that non-coding sequences, that not only exons are coding for amino acid, intron do not code for amino acid. Intron, enhancers, promoters, regulatory sequences, microsatellite, many satellite, VNTRs, variable number tandem repeat. Likewise, there are so many. Okay, so now let me read the text sentence. I just give the primary information here. Now let me show you the text sentences. You can understand, I think so. So what is the second approach? That sequencing the whole set of genome, sequencing the whole set of genome that contain, sequencing the whole set of genome that contained all the, why I am spending this much time? Because these are the key line which we are going to get in examination, that contain all the coding and non-coding sequence. Should remember sequence annotation, because there is no choice for the examiner other than these particular lines. We have only two major approaches, we said. One is expressed sequential tag, key line, identifying all the genes that are expressed as RNA, and other is sequence annotation, sequencing the whole set of genome, please excuse me here, whole set of genome that contain all the coding and non-coding sequences, that we call it as sequence annotation. Later, these particular different reasons in sequence with function, later assigning different reasons in the sequence with functions. First of all, coding. Then after only they will make the differentiation of this particular that coding and non-coding, who is doing function, who is not having function. So now you may get doubt here. Do any nucleotide base sequence without function in our DNA? Yeah, 97% of our DNA is junk DNA. So that is one of the approach. So we are just following these two issues here. That's why let me go for this expressed sequential tags and sequence annotations. That is okay. Now let us see what is DNA sequencing. How do the DNA sequencing has done? How do the DNA sequencing? What is DNA sequencing? And how does it is done? DNA sequencing is the process of determining exact order of the 3 billion nucleotide base pairs. Exact order of the 3 billion nucleotide base pairs, that adenine, thymine, guanine, cytosine, which make up the DNA of the 24 different chromosomes. 24 genome of male is 24, isn't it? And 24 in the male, 23 in the female. That reading total 3 billion nucleotide base pairs, how they get arranged, ATGC, TACG, likewise the multi combination, the sequencing like this. I just wrote 4. You have to imagine the total 3 billion base pair, 3 billion base pair. Okay, so that you have to remember. Okay, so sequencing all that paired chemical building blocks, we say nitrogenous bases, adenine, thymine, cytosine, guanine, that make up the DNA of the 24 different human chromosomes of the male and 23 in the female, was the greatest technical challenge in the human genome project, because not a joke that this much number of nucleotide base sequences identifying. So what are the approach is during the DNA sequencing? For the sequencing, the total DNA from a cell is isolated fast. In the cell, that particular nucleus contain this particular chromosome. First of all, we should isolate them, isolated, converted into random fragments. First, we isolate the DNA and we convert them into random fragments, simply in our language, cutting into pieces. Random fragments of relatively smaller size. Take a newspaper, cut the newspaper into 1 lakh pieces. There you can understand what we are doing in Human Genome Project. Okay, that's not the homework. The cutting the newspaper, just saying like so. So that means making the random fragments by using what? By using, we have that restriction endonuclease enzymes. Do you remember the molecular biology? We learned molecular base of inheritance. There in the chapter itself, we learned here. Actually, this is also that particular part here. That last part of the molecular basis. Why I'm saying you did you hear? Because molecular basis of inheritance said by botany, sir. So that's why I'm saying restriction endonuclease is a recombinant DNA technology used. The same restriction endonuclease enzymes we are using here for cutting of this particular DNA into many fragments here, that random fragments, which are very smaller in size, and cloned in a suitable host using specialized vector. So these particular fragment, which contain some nucleotide base pairs, which are taken from human, will be either introduced into a suitable vector. Let me make Escherichia coli. I introduced this particular into Escherichia coli. Let me take another color to make differentiate. So this is the piece, and I introduced into the Escherichia coli. That you may know that PBR 322 plasmid. With the help of that, we introduce into the another vector. Okay, so the cloning results in the amplification of the DNA fragments, which are used for sequencing the bases. Okay, and that means that the Escherichia coli, I took Escherichia coli, introduced them, and I multiplied this particular bacteria. When I multiplied, this copy will be multiplied into thousands and lakhs, isn't it? So that's it. That means making in the random fragments and introducing into a particular vector. Generally, what we use as a vectors, bacterial artificial chromosomes. We have to say bacterial artificial chromosome. Next, yeast artificial chromosomes. The commonly used hosts are bacteria and yeast, and the vectors are called bacterial artificial chromosome and yeast artificial chromosomes. Okay. The fragments were sequenced using automated DNA sequencer. That was a big task actually for the scientists. So that means simply, let me say, I said you to cut the newspaper into 1 lakh pieces. So that each and every fragment you are reading, actually individually are reading. So after all cutting this particular newspaper into that 1 lakh pieces, do you able to see this, but this particular part belongs to which piece of the whole newspaper? So let me talk the same. If you cut this particular DNA into random fragments and pieces, after all knowing the sequence of this particular DNA, can we identify where it is exactly in that particular thread, that means in the total genome, That is a tough task. That a lot of time has taken over for that itself, for a longer period of time. That was a bigger task to the scientists at the time when they're doing genome project. Reading is so matter that as nucleotide based sequences, but location of that particular nucleotide pieces, the cut fragment in the original genome is going to be a tough task to the scientists. This problem is solved by these fragments are sequenced using automated DNA sequencers that work on the principle of method developed by, you remember him, Frederick Sanger. Sanger-Coulson sequencing, we use it to say in general. Our text is given only Sanger, Frederick Sanger, that the fragments are sequenced using automated DNA sequencers under that the technique developed by the Frederick automated. That in our manually that by human being is not at all possible here. Not at all possible. Automated DNA sequencers introduced by the Frederick Sanger. After that, this alignment is possible actually. Humanly, this kind of alignment, that means with each and every fragment, after cutting into pieces, 1 lakh pieces, the newspaper, is it possible to you to make attach again? If I ask you, attach all the papers and come tomorrow, otherwise I don't allow you to into the classroom. Is it possible? Humanly not possible. But that DNA fragments which we are cutting into fragments after all reading into the sending into the vector and after all studying them, the sequencing of that is possible in automated DNA sequencers by Franger, so the Frederick Sanger-Coulson sequencing usually say in the field of genetics. That alignment of the sequences is not humanly possible. That you have to remember. So therefore, that for that what is developed? Specialized computer-based programs were developed. Supercomputer required. After all, the identification of supercomputer only, that means automated DNA sequencers by using supercomputer only, this is actually complete. Advancement in the technology, I said to you before 2005, targeted 2005 but completed in 2003 because of that supercomputers. That what we said bioinformatics. So these sequences were subsequently annotated and were assigned to each chromosome that by with the help of these specialized computer-based programs only. The latest method of sequencing. Wait a minute. You got a small disturbance. Not a that see the particular reading that particular DNA fragments is not a bigger problem. That these particular DNA fragments to be assigned an original that genome, where it is exactly in the original genome is a bigger problem. As we said now, automated DNA sequencers, and now the latest method of sequencing even longer fragments by a method called shotgun sequencing using supercomputers replaced the traditional sequencing method. So as long as the technological advancement is coming, our process became much more easier. Okay, so to be remembered, to be frank, till the time we are completed only 4 issues. One is the major approaches. One I said that the sequence annotation I said, and that can anyone help me? What I said the another one? Sequence annotation, major approaches of the Human Genome Project, sequence annotation, and so that's what. Here the lengthier issue it is. Lengthier. When we go for explanation, we can go for hours, but we should not miss the content which can come in examination. Expressed sequential tags. Expressed sequential tags, sequence annotation. That is one of the major approaches we said. Then after that assigning that how do we do the genome project, a brief information is given that in that, as I said, we cut them into the random fragments we cut, and that we introduce it into the bacterial artificial chromosome and yeast artificial chromosome. As like they create the bacterial artificial and yeast artificial chromosome by introducing them into the vectors. We call them as bacterial and yeast artificial chromosome. And random fragments reading is not a bigger problem. that making them in the original DNA, that means their original position in the DNA is going to be a bigger problem. That is solved by Frederick Sanger. And that what do you say? That Frederick principle of a method developed by the Frederick Sanger help whether the fragments are sequenced using automated DNA sequences, and more advancement in the technology, shotgun sequencing, that using the supercomputers is also one of the technique. This is the issue we are seeing. Okay, this is the technical information. Now, what is there? Okay, that 13 years, that paid effort. So, what you came to know? What you came to know of this particular human genome? Salient features of human genome. Okay, you read for 13 years. Do you say something to me? Yes, we have. We can say only something to us based on our text. Okay. So, what is the that information you got after all reading? What do you know about Human Genome Project where Z completed? What is the information? There's some information we are going to learn here. Human genome consists of chromosomes made up of nucleotide base pair that we know. Okay, then the average gene consists of 3000 bases. Average genome consists of 3000 bases. Every gene consists of 3000 bases. Minimum. We are not going to say maximum. Sizes vary greatly, with the largest known human genome, human gene being one of the longest one. Gene for dystrophin is the longest one actually. Dystrophin gene. Do you remember this dystrophin gene? Deficiency of this particular mutation in this gene only cause a disease called X-linked disorder, Duchenne muscular dystrophy. Okay, this is the longest gene actually. Okay, so the total number of genes is estimated as 30,000. 30,000. Total number of genes in human estimated as 30,000, and 99.9%. 99.9% nucleotide bases are exactly same in all the people. Just remember, this will be helpful for us in DNA fingerprinting. 99.9% of the nucleotide bases are exactly same. The functions are unknown for 50% of the genes we discovered. Still, we don't know what function they are doing. 50% of genes out of the discovered, that their function is still unknown. Okay, less than 2% of the genome code for proteins. Protein coding genes are less than 2%. Exact number to say 1 to 1.5%. So less than 2% of the genome code for proteins. Excuse me. Next, repeated sequences make up very large portion of the human genome. Repeated sequences are going to be that in the repeated sequences, so many different are there, clustered repeats, dispersed repeats, likewise. They make up large amount, repeated sequences. Simply in normal language, we can say the sequences are much more times repeated. They occupied that repeated waste is so much in that genome. Junk DNA, that's why I said. The repetitive sequences are stretches of DNA sequences that repeated for many times. They are thought to have no direct coding activity. No direct coding activity they'll have. No direct coding activity. Lost my voice. Okay, okay. 3000 bases, dystrophin gene. That means why I'm writing only keywords. By looking towards them, you have to say. So let us see one. Take a look on that 3000 bases. What is 3000 bases? The average genes, average bases per gene, which identified in human genome. Dystrophin, longest gene, which contains a large number of nucleotide bases. 30,000. What is 30,000? 30,000 genes in human. 99.9%. What is 99.9%? Which that most of the that all for all human beings 99.9% is of the chromosomes are going to be same. 50% of genes, the 50%, what is 50%? that 50% of the genes are known till the time their function is unknown. We discovered, we sequenced them, but their function is unknown. Okay. Next, what next? 2%. What is 2%? The protein coding genes are less than 2%. Next, repeated sequences. Most of the human genome contain the repeated sequences. And these repetitive sequences are stretches of DNA sequences that are repeated for many times. They are thought to have no direct coding functions, but they shed light on the chromosome structure. They are not exactly having function. That protein coding function is not there, but they are important. In what way? To make up the chromosome structure and dynamics, and to understand the evolutionary process, that repeated sequences are much helpful for that. Next, chromosome 1 have chromosome 1 is having 2968 genes, which is the maximum. That particular first chromosome is top in the having genes, and Y chromosome, gene for Y chromosome, genes on Y chromosome are 238 gene, which is the minimum number, maximum, minimum. We are writing. So scientists are identified 1.4 million locations where single DNA differences, we say I am writing here, single DNA differences, we say single nucleotide polymorphism, which are pronounced as SNPs, single nucleotide polymorphism, single nucleotide polymorph sequences. We can say SNPs. What is this exactly? These are 1.4 million locations where single base DNA difference means that if you see a sequence like A, T, G, C, something like this, there is a sequence. Okay. Somewhere else, the similar kind of sequence is also there in the chromosome, in the genome somewhere else, with a small difference like this, A, T. Such kind of locations are 1.4 million sequences we are talking. Do you find any difference? Yeah, we have only single nucleotide, this area, that different from here. Single nucleotide base difference we can see. Okay, see that this is the only difference here. These kind of locations are 1.4 million nucleotides. This information promises to revolutionize the process of finding chromosomal locations for disease associated sequences and tracing the human history. Okay, so that's it. This is the thing, and I do explain once again because these are whatever they are on the board, they are very important for us. Okay, 3000 bases. It is said to be that average number of nucleotide base pairs in each gene is going to be 3000. Dystrophin, that gene, that dystrophin is the protein, that gene is having large number of nucleotide base sequences, 30,000. Said to be 30,000 genes are present in the human genome, 99.9%, which means is going to be same in all. The genome of 99.9% of the genome is same in all the people. Then 2%. What is 2%? 2% is the protein coding genes are going to be less than 2%. Repeated sequences. That means most of the DNA sequences are going to be repeated sequences. The clustered repeat, dispersed repeats likewise. Just remember as repeated sequences. Then first chromosome is having 2968, Y chromosome is having 238 genes, and 1.4 millions of the nucleotide base pairs, which are 1.4 millions of the locations are going to be with single nucleotide base sequences. Okay, so along with that, I'll repeat before I close this, I'll repeat that earlier learned the two major approaches: expressed sequential tags, sequence annotations, and bacterial artificial chromosome, yeast artificial chromosome. Means what I'm saying here, what I'm saying, where to be focused, that's all, where to be focused over, that's what that area. Okay, because we have less information, but from here we are getting that always we are getting question from here. Sanger course that Frederick Sanger, Sanger-Coulson sequencing, and these are the things to be remembered. This is the area on screen, whatever that particular I'm showing, that is going to be question. Remaining all are general topics we can see. Then advantages of human genome project. Advantages of human genome project. Okay, let's talk about the advantages of human genome project. That in the area of healthcare, identification and mapping of the genes responsible for genetic diseases, help in diagnosis, treatment and prevention of these particular diseases. So simply we call health purpose. So advantages in the healthcare, identification and mapping of the genes responsible for to identify various genetic diseases before the birth itself. We can diagnose, we can read, and we can create the reverse mutations. That normal condition, you may have learned over hemophilia. This is normal female, this is hemophilic female. What nucleotide base sequence make them like this normal? What nucleotide base sequence makes that particular female to be hemophilic? If we know, a hemophilic mother can give birth to the normal son by making reverse mutation where the changes are happened, we can identify. So that's why that very good in genetic disorders, we can say. Next, detailed knowledge of the genomes of humans and other species will give a clearer picture of gene expression, cellular growth, and differentiation and evolutionary biology. Cellular growth, why some cells continuously divide, why some cells will stop the division, what genes are responsible for that, why some cells will go quiescent for a longer period of time, then how do they get activated? We will get answer for cancer or that more other diseases and various kinds of old age reasons. Something simply we can say that why we are getting old age, what genes make up. So if we correct all of that, that in future we can increase our lifespan also. And earlier detection of the genetic predispositions to disease, rational drug design, gene therapy is going to be easy with more knowledge on human genome. Okay, means for example, I'm saying here, we learned about the bald head, isn't it? Bald head. So a person is having these particular dominant genes, he's not by birth bald head, isn't it? Means over a period of time when he get 30 or 40, nowadays 25 even, because of other factors, pollution or something else, so he'll get bald head. Okay, so what we have to do, earlier detection of the genetic predispositions to such kind of disease, the father is having, definitely he'll have, and will study that particular, will make it correct. So correction is not like that writing directly from capital letter to the small letter. You have to understand there is a lot of process, but simply for your understanding, I am saying. Okay. So a new era of molecular medicine characterized by looking into the fundamental causes of the disease, then treating the symptoms will be an important advantage. What do you say? Molecular medicine. The future is about this only, molecular medicine. Right, directly root cause of the disease will go for why the disease is coming into this area, why to these particular people. Nowadays you may have a discussion even with coronavirus, isn't it? They are studying even genes. Why? Because in some areas it is spreading wild, but in some areas it is spreading very little. Why? What makes that? Why Indians are less people we are dying in India? So that all they are studying. In some other countries you may have seen over, because virus attacks, people dies. Virus attack, people dies. There is a country, I think Chile, that 80% of people are going to that death rate is 80% nearly. But our death rate is one less than one. Why? Because some genetic makeup, what genetic makeup is going to be tolerating that? That kind of issues can be studied with the genome project easily. Okay, it's a vast area. Okay, so today in the classroom, I just restricted myself to the text. Tomorrow I'll give that project outline. Tomorrow I'll give the project outline. In the next class I'll give, and then after we'll go to the DNA fingerprinting. But here I should that while explaining, I should take the consideration of text also, because a very interesting thing here, that very small area we are getting questions. This is the area. Nothing to worry. Just read the text and go through these particular words. If you are able to answer, you will get 4 marks easily. That means for one question carry 4 marks in Neet, isn't it? So that very small area to say, very small area. Technical words. I say I made that words in the keyword method. I will showing here. So if you read this, I'm not going to say that only word will get. That means based on this word, you have to learn the content, or you check yourself whether by looking this particular, am I able to explain or not. So if you do practice likewise, take the screenshot of this and you practice, you read the text. If you have any doubts, you ask. And we just go for a little general information about the genome project. I have a chart which I prepared, that I'll show the chart, and there I'll show you what are these exactly that genome project, how, what kind of different kinds of sequences are present. All that issues we'll discuss in the next class. Okay. So then uh, but this is the primary importance. This is the primary importance, you should remember.